So this video is on the lab 7, which is on buffer solutions in the blood. And so in this lab, you will review the definition of a buffer solution, observe the effects of strong acid on proteins found in blood serum, and learn how to prepare a buffer solution and observe the effects of strong acid on the buffer solution and also of a strong base. It's not saying that here in the introduction, but you will also see how when we add a strong base, you know, how the buffer is affected by that. So it's really all about buffers. And then here there is some background information that talks about the human body. And so to function properly, humans need to have a blood pH maintained between 7.35 and 7.45. pH for the blood below 7.35 results in a condition called acidosis, and there are symptoms for that, and a buffer above 7.45 is called alkalosis, and it's also big problems, health problems ensuing that, and can even be fatal in both cases. So that's why we need a buffer in the blood, and it's an important concept as you want to go into the nursing field to understand the role of buffers. And so the reason why the human body has a buffer is because metabolism of proteins, starches, sugar results in waste products that must be excreted from the body. And so these waste products are transported through the blood to the kidney. And because some of the waste products are acidic, a buffer system is necessary to maintain the blood pH. For example, uric acid is a waste product from the metabolism of protein, and lactic acid is also produced during the metabolism of your muscles. So they go into the blood, they could make your blood acidic, and this cannot happen because this would jeopardize the nature of the proteins that are necessary for your body to function properly. So that's why the blood has a buffer to maintain the pH around that narrow range of 7.35 and 7.45. And so that's what a buffer does. It maintains the pH at a given value, and the value depends on the buffer itself. By definition, a buffer solution requires a weak acid and its conjugate base to be both present in significant concentration. So that's what, in chemistry, that's the definition of a buffer that you want to remember. For a conjugate acid-base pair to be a buffer, and you need both the acid and the base, the acid needs to be weak. So all words are important here. The acid has to be a weak acid. And then the conjugate base, it has to be the conjugate base of that acid, right? Not any conjugate base, but the conjugate base of that acid. And they both need to be present in large amounts so that they can, do their job of buffering. And so the human body has several buffer systems, but it uses mainly the carbonic acid and bicarbonate, bicarbonate ion to make a buffer in the blood. And so why this one? It's because it's convenient, because carbonic acid naturally exists in equilibrium with water in the bloodstream. And so that's carbonic acid. H2CO3 is the formula for the carbonic acid. And carbonic acid is unstable when it's in water. It easily dissociates into carbon dioxide and water. So it's in equilibrium in your blood, especially because CO2 is a waste that we produce and that needs to be eliminated. And it's also going through the blood, you know, up all the way to the lungs and then exhaled. But so because we have some CO2 in our blood, you know, because it's a waste we need to expel, then with the water from the blood, it's making the carbonic acid, and so that's readily available. Being a weak acid, carbonic acid partially ionizes and then dissolves to produce the hydronium ions and the conjugate base, the bicarbonate. So the carbonic acid, when it's placed in water. So this should remind you what we did yesterday. So we have here an acid because it starts with an H in its formula, and it's even what we call a diprotic acid because you have two hydrogens in the formula that are written first in the formula, so that suggests two acidic hydrogens placed in water. And so water will play the role of a base here because this is an acid. So water will accept a proton from the carbonic acid when carbonic acid donates its proton. So when H2CO3 donates a proton, so you remove an H plus, so remove an H plus, then there will be only one. Oops, there's a mistake here. It should be H. Let me. Yeah, that's a pity. We have a mistake here. I'll have to review the documents. So when you remove an H, so the subscript 2 here is removed because you have only one hydrogen now. And then when the H leaves with its positive charge, it leaves behind a negative charge. And here it's there, it's tiny. I think I'll change it to something like that. So this is the conjugate base of H2CO3, right? When H2CO3 donates a proton, it becomes HCO3 negative 1. And then water will accept the proton as a base. And so water plus H plus. So that means you will have 3 hydrogens. So then it becomes H3O. And then the hydrogen came with a positive charge. So it's showing up there. Okay, so that's the conjugate acid of water. So it's acid here plus base here gives the conjugate acid of water plus the conjugate base of the carbonic acid. And so we have this in this equilibrium happening in the blood. And yeah, so since the equilibrium constant for this reaction is approximately 3 × 10⁻⁴ here, it's a -4. Only about 1 out of every 3,000 carbonic acid molecules ionize and become the conjugate base. So it's a weak acid. So remember what a weak acid, it means it partially dissociates. And the proportion of ions is very small compared to the proportion of the acid plus water. So you have a lot of the acid and very few of the bicarbonate. And so because of that, to have a buffer, we need as many bicarbonate as we have the acid for this to work as a buffer. So then the bicarbonate ions are released into the bloodstream by the kidneys. So the kidneys also are involved in this and release the bicarbonate ions so that there is enough in the blood to, make the H2CO3 and HCO3 negative 1 a buffer for the blood. So that's the, background information here. So this lab again has videos, and here there is the playlist. It's this South Seattle professor who has done those videos. And so, so the first part of the lab, part A, it's more like a theoretical work here. You need to select, for question 1, select out of these 3 pairs, so H2CO3 and CO2, NH4 plus and NH3, or H2CO3 and C2H4O. Out of these pairs, which pair do you think can make a buffer? Which one will be working as a buffer? So remember, go back to what is the definition of a buffer, what a buffer requires to answer that question, right? It needs to be a weak acid and its conjugate base. So find the one pair that is made of a weak acid and its conjugate base. Then question 2, write the chemical formula of the conjugate base of each of the following compound. And so here you have 2 formulas, and you need to take these as, so we need to find the conjugate base. So that means these are acids, and you need to find the formula that they become when they donate their proton, right? So these are your acids. They will donate a proton and become the conjugate base. So find the formula when you remove an H plus from this formula. So don't forget, it's removing an H and a positive charge. So what I mean here is don't forget to look at the charge. The charge may change. Question 3, it's the reverse. You need to find the chemical formula of the conjugate acid of these. So conjugate acid, which means these are your bases. These are bases, and you need to find the conjugate acid formula, which means what do they become when you add an H plus to them? Because as bases, they will accept a proton. So they will accept a proton and become the conjugate acid. So add an H plus to those and find out the conjugate acid formula. So it's what we did in the review of chapter 9, right? It's the same process. So that's part A. Part B, effect of an acid on a serum protein. And so part B is short too. It's just this. So this will require that you look at this video. So the video will show you an egg white. Why egg white? Because egg whites contains a lot of albumin protein, and this can simulate the proteins found in the serum, because a lot of proteins that are found in the blood are albumins. So here, blood serum contains dissolved protein called albumins that transport molecules such as fatty acids and hormones that would be otherwise insoluble. So we need those proteins, to transport fatty acids and hormones, that need to be delivered elsewhere in the body. And so we need those albumins to be able to do their work, right? And float in the blood. So they should not be changed, right? So here you will see, add an acid to egg white, it will change it. And so we want you to write the observation. So to be able to understand the change, you need to first write your observation of how an egg white looks like before you add the acid. So just describe what it looks like. So I want to emphasize here, because I think some of you still have confusions about what clear means. Something clear, like a clear solution, means it's transparent. It doesn't tell about the color. It only tells about the transparency of the solution. So egg whites, are transparent, but they are viscous. They are not that liquid like water. So that's what clear means. And then you will see that they will change and become white. And so if if they become white and you cannot see through them, through the egg whites, that means it's not clear anymore, right? So don't use clear anymore after that. But there's a difference immediately after the acid has been dropped in. And then there's even more difference when you let some time for the acid to act, and then 5 minutes after it's, even more changed. So this changed the physical properties of the egg white, right? So that's what your observations should be. You don't. So I've noticed also people tend to make some inference when we ask for observations. Sometimes students write what does it mean, what they conclude from the observation, but that's not what an observation does. An observation is only telling what you see, what you feel, what you if you can touch or smell, it's all these um, things that you can feel, right? Or see or hear. And so question 4 is there for you to do the inference. So using the observations in table 1, extrapolate and predict the effect of an acid pH on the blood of a person. So what would happen in particular to the serum proteins of the blood when you are adding, when you have a acidic pH in your blood? What would happen? So that's part B, effects of acid on serum proteins. So part C, it's a much longer part here. That's the bulk of this lab. And here you will observe the preparation of a solution that contains the same buffer system found as in the human blood. And so that buffer system is made of carbonic acid and the bicarbonate ion, right? Together they make a buffer. And so carbonic acid, because we've seen that CO2 is in equilibrium with carbonic acid when it's placed in water. So you will introduce the carbonic acid by exhaling into the solution, blow into the solution. And that will introduce some CO2 in the solution, and the CO2 will convert into carbonic acid because they are in equilibrium, CO2 and carbonic acid. And then the bicarbonate will be added as a salt. So sodium bicarbonate is the salt of sodium and bicarbonate bonded together. It's an ionic compound. This will be added to the solution. So sodium bicarbonate, if you don't know yet, it's baking soda, right? Baking soda is sodium bicarbonate. So you will make your solutions this way, or watch the video that shows how this is made. And there will be a table, and I can, I can move here. This will be the table. Table 3 will be your observations. It's actually this video. It's your observations when the buffer solution is prepared. So you will notice the volume of water that is used, pH of the water before we blow the CO2 into the water. What happens to the pH when the CO2 is exhaled into the solution? So here we expect words. pH of the carbonic acid solution. So after the professor has blown into the solution for 2 minutes, the pH will change, and so the new pH needs to be entered here. Mass of, and then once we have the carbonic acid in the solution, then we want to add the bicarbonate ion. This will be added with the salt or the baking soda. So mass of the baking soda, this is a solid, so that's why you can take the mass. And then once it's dissolved, then the final pH of the buffer solution. So final pH of the buffer solution. So that's all about preparing the solution. And the solution will have to be prepared twice because then it will be tested by adding acid to it. That's this table there. And then it will be tested again by adding NaOH to it. So solution will have to be prepared twice. But before that, she also uses an indicator. And so it's here again. It says, so two methods will be used to monitor the pH of the buffer solution. First, an indicator made from butterfly pea flower tea will be used as a visual indicator of pH. So it will give a color to the solution, and the color will tell you if you have an acidic pH or a basic pH. And then the second method will be using a pH probe, a pH sensor that reads electronically the pH value. So the probe is a chemical system, but then it translates into a electrical signal that is then converted into a pH value, and so that will read directly the value of the pH. So this first video actually is to show you the effect of the pH on the color of the butterfly pea flower tea. So there is that, tea that is used to change the color of the solution. And so you will watch that video. And so here observations, it doesn't specify exactly, but I would like you to tell what is the solution. So, is it just water, or is it a solution of maybe carbonic acid or something? So tell what it is, the name. You will see she reads the pH with the probe, so you can have a electronic value for the pH. And then observation. So this is mainly what is the color. So be precise about what you see for the colors depending on the pH. So you will see that depending on the pH, if it is acidic, it will have a different color than if the pH is neutral, than if the pH is alkaline. Alkaline means basic, right? It's synonymous to basic. So pH indicators with color like that can be very useful when you do some what we call a titration of an acid by a base or vice versa. So that's the first table here. Then you will see the preparation of the solution, and we've been through this. That's the second video. Once the solution is ready, the carbonic acid bicarbonate buffer solution, then we will add a strong acid to it. So HCl is a strong acid. It's going to be added, and for comparison, it's going to be added to water and to the buffer you just prepared, the carbonic acid bicarbonate solution. So you need to, so that video lasts a little longer because a lot of drops needs to be added on both beakers. So one beaker contains, so actually water is on this side, and the buffer H2CO3 and NaHCO3 is on the left here. So make sure you write your values in the proper column. So you need to read both at the same time, right? Whenever she had drops, she had those drops, 2 drops in each beaker. So you need to read each values at the same time. And so you will see the pH will change. And so here we are adding an acid. So the pH will decrease, overall for both, the pH will decrease. But, a good buffer should maintain, and that's the role of buffers, that's what we expect from them, it's that it should maintain the pH in a narrow range, right? That's what the buffer in the blood should do. It should keep the buffer between 7.35 and 7.45, which is a really narrow range. Here, that buffer will not be able to keep the narrow range like that, but it will be much better than no buffer. You will see that, right? Water is not a buffer. So she will stop adding the drops in both and show the pH value at 22 drops here. But the video doesn't stop there. After that, the professor, she's trying to show you how many drops you need to add to reach what we call the buffer capacity. So what is the buffer capacity? So a buffer is able to maintain the pH, like for example, the blood buffer is able to maintain pH at 7.4, in that narrow range. And but it can do that as long as the amount of acid or base, and here we are testing the acid, as long as the amount of acid added, the extra acid added to it to test it, is not too large. If you add too much of it, at some point the pH will, have a dramatic drop more, and be very, very acidic, right? So not anywhere near the starting value. So that's when you have reached the buffer capacity. When so you will not graph that part, but you will stop at 22 drops for your graph. But at some point, if you were to continue and get the values, you would see a more dramatic drop in the pH, and that shows you that the capacity of the buffer has been exceeded. The buffer is not able anymore to maintain the pH. And for water, which is no buffer at all, it should be, as soon as you add a few drops of acid. So in that table here, total number of drops of acid required to reach the buffer capacity of water, it should be a very small number of drops. But the total number of drops of acid required to reach the buffer capacity of carbonic acid and bicarbonate buffer, this should be a number that is larger than 22, because here the previous table stopped at 22, but that's because we cannot go on and on. So she adds those drops more quickly to try to see when you are reaching the buffer capacity, and it's really a lot more drops. So this should be a much larger number, right? And that tells you the bicarbonate and carbonic acid buffer is acting as a buffer because it's able to maintain the pH. So then you will need to graph what you have in this table 4 here. All the data from table 4 will be graphed. The drops of HCl will be your x-axis, and the pH values, and you will have two curves. The pH values will be in your y-axis. And so there are different ways you can do this, and you are provided a graph in Excel, and you're provided a graph on paper if you prefer to print and do it by hand, right? So let's look at this. So it's not in that document. You will need to go into Canvas. Where's Canvas? Sorry, I'm lost. So here I'm showing you the Canvas page, right? Our Canvas page where you have the lab 7 buffer and acid bases. So as always, you have the documents in Word for the lab report. If you like to type in your answers, and I would, recommend that. This time you can use that. There is the PDF if you prefer to print, and that's fine too. And so here, the graph template for printing. So this PDF, why is it? Oh yeah, PDF, sorry. Sorry, yes, it has to be a PDF because that's something you would print. So let me show you what it looks like. It's this. So it's this, just a blank page with a graph. That's it. So, it shows you exactly what you need to have for the x-axis. So you will add, drops 2 by 2. So that's why it's only the even numbers, and it goes up to 22 drops in total. On the y-axis, it's the pH value, and pH typically will go from 0 to 14. And you will see that actually we could have narrowed that down. So on Excel, you will be able to narrow that so that it shows your curves more in details. And then the title is even added for you. So it's all ready for you to use. And so the only thing you need to think about is that for the two curves, it would be nice to have two different colors, or two different symbols for the dots that you add, a cross or a dot, but make them different so that we know which curve is which. And then you need to label. That's important here. Label each curve. This is not prepared for you. You need to label each curve and say this curve is that, and that other curve is, the pH of water, and the first curve is the pH of the carbonic bicarbonate buffer. So that's if you want to print, you would use this. If you want to use the Excel graph and you are at ease, typing the values of pH in the Excel graph, then you would use this one, XLSX file. So let me show you what it looks like on Excel. So here I'm sharing the Excel file. It's this one. So it's prepared already for you, and you have instructions on how to do the graph. So first you would need to type, the pH, and I don't know, let's say it's 6 point, so water, I don't know, 6.01, whatever. You type your pH values here, and then follow the instructions on how to do the graph. And I would recommend to do the two curves in the same graph. So do the step 4 instead of create a second graph. One graph for the two curves would be better to compare. So you need to select the columns when you do this. Select the columns or select data like that. And then insert a graph, scatter. Right. So read the instructions, right click. Okay, so if you're at ease with Excel, use that. But if you prefer to print, of course, those who cannot print, they will have to use this. So if you need help on this more specifically, let me know. So that's the Excel file where you will do your graph. And so once you have your Excel graph, you can insert it here. If you, for example, when you're on Excel and you click on, select the graph only, then you can copy the graph and then paste it here. So that's one way to do it. If you make a picture of the printed graph, you can also insert it here. Or you can have it at the end of the document if it's too complicated to insert it here. It's fine too. Okay, so you will need to do the graph of the addition of HCl into the water and buffer. And then interpret your observations and analyze the graph. So here there are questions about, did the water or the buffer solution act as a buffer? So tell, which one of the two Is it the water or the H2CO3 NaOH solution that acted as a buffer? And question B is basically asking, what behavior observed during the experiment indicates a solution acts as a buffer. So what makes you think the solution you said here is acting as a buffer? What's particular about it that tells you it's a buffer? And then question C, compare the number of drops of acid required to reach the buffer capacity. Compare, the number for water and the number for the carbonic acid bicarbonate buffer. So those two numbers here, that's what you want to compare here in question C. And as I said, one should be a lot more than the other. And that tells you which one is the buffer. So that's for the acid. And then you will do the same thing for actually you will not do a graph for the addition of sodium hydroxide. So sodium hydroxide is a strong base, right? It's a strong base because it has the OH minus ion here. The hydroxide ion is a strong base. So instead of adding an acid to the water and buffer, we will add the strong base to the water and buffer. And so there is this other video, fairly longer too, because, there's a need for a lot of drops to see the buffer capacity of the H2CO3 NaOH solution. So same thing, except that here the pH should increase, right? Because when you're adding a base, a strong base to a solution, a basic pH is a pH above 7. So you will see the pH increase slightly as you, add drops. Well, slightly if you have a buffer, but dramatically if you have no buffer, right? Like water is not a buffer, and you should see a sharp increase right from the start. So that tells you there is no buffer there. So again, the video doesn't stop when the professor has added 22 drops in each beaker. She needs to add a lot more to reach the buffer capacity of the carbonic and acid and bicarbonate buffer. So again, here the total number of drops of base required to reach the buffer capacity of water should be a very small number. And here the total number of drops of base required to reach the buffer capacity of carbonic acid and bicarbonate buffer should be a number much larger than 22. So all the way to the last drops that she added to reach the buffer capacity. And again, buffer capacity is reached when you see the pH, in this case, increase sharply, right? When there is the pH is not maintained anymore, and it's increasing sharply because you add a strong base. And so you will see that the buffer is better at maintaining pH with acid or base. You have to tell which one it is better at maintaining pH. Is it when you add acid or is it when you add a base? And why? So why is it better? So think about the definition of a buffer. What do we expect from a buffer? It's gonna be better if it does that better. So maintain the pH. And then question 8, you need to write the chemical reactions that occur in the blood buffer. So again, H2CO3 and NaHCO3, the carbonic acid bicarbonate buffer. So what happens when NaOH is added? So that's what a buffer does. When you have a buffer, you need very large quantities of both the weak acid and its conjugate base. And the reason is because it needs to be able to buffer it, whatever you add to it. And when you add a strong base like NaOH to a buffer, NaOH being a strong base, it will react with the acid of the buffer, right? So in the reactant here, the A and B, so one of them will be NaOH. So NaOH will be either A or B. And then the other reactant has to be the weak acid, because this is a strong base, so it will react with the weak acid. So you want to have the weak acid, as A or B here. And then you do the transfer of proton from the acid to the base, and you will find the conjugate acid and the conjugate base, which are C and D here, right? So that's how you want to do the chemical reaction. And so same thing when you are adding HCl. So this time you're adding an acid, right? HCl is an acid. So when you're adding an acid to a buffer, the acid that is added will react with the conjugate base of the buffer. So that means it's NaOH that this time that will be your reactant with the HCl. So both HCl and NaHCO3 will be reactant. And again, to complete the reaction, you need to do the transfer of proton from the acid. So HCl will donate its proton to the base, and NaHCO3 will accept the proton. So there's two ways actually to write those chemical reactions, because you could drop the Na here, Na plus, and not have it. Or you could drop the Na plus here again. Na plus in this case is what we call a spectator ion. It does not, it stays the same before after, except it combines with the negative ion. And so there's two ways to write those. You could drop the Na plus and just write OH minus. Don't forget the minus, right? OH minus. And here, when you use NaHCO3, you could also drop the Na plus and write HCO3 negative 1. But you can keep them, it's fine too. So it's just that Na plus is a spectator ion and could be omitted from the reaction.